So far we discussed about photosynthesis, primarily the light reaction. Okay, so now you should be very familiar and very comfortable with understanding where do you get the energy from. Okay, and this is going to be solar energy, most of it, light energy that gets converted into essentially chemical energy stored in the form of ATP and NADPH. Okay, so now today we're going to talk about CO2 fixation. Really we call them CO2 fixation, meaning CO2 is a gas, right? CO2, this guy normally is a gas. So we fix it, okay? Fix it, meaning that we turn gas into some kind of organics, okay? All right, so that's what it means by CO2 fixation. Now, before I move forward, you need to understand that there are actually many different types of CO2 fixation pathways, okay, in biology. But primarily, we're only going to talk about the Calvin cycle, okay? We're only going to talk about this Calvin cycle because this is the major pathway for photosynthesis, carbon fixation, okay? The major pathway for the dark reaction, okay? But there are other bacteria or archaea that have other pathways, okay? For example, you guys can imagine, okay, you guys understand Tca cycle, right? What does Tca cycle do? Converts acetyl-CoA to what? CO2, okay? So acetyl-CoA to CO2, and then you produce what? Tca cycle produces energy, okay? Energy in the form of ATP, NADH, and FADH2, right? So you can imagine, okay, Tca cycle is a pathway that converts organics to gas, and you take out energy, right? So in this world, there is a pathway called the reverse Tca cycle, okay? So you just put back all the energy you get, and then you can fix CO2 and make acetyl-CoA. Okay, so there is such a pathway in some selected microorganisms. Okay, but what we're going to be discussing is not those. Okay, we're going to be discussing the Calvin cycle because Calvin cycle is the most abundant pathway that occurs in plants, in algae, in cyanobacteria. Okay, and Calvin cycle really accounts for most of the organics that you find. Okay. So, without further delay, this is what the Calvin cycle looks like. Okay, it's a complex cycle, but we're gonna go through this in detail, right? I really like this particular picture of the Calvin cycle. So if you have the textbook, you will find that the textbook usually just say, okay, you have ribulose biphosphate becoming ribulose 1,5-bisphosphate, blah, blah, blah. But a lot of times they don't show you how many times you have to do it. Okay, so this picture actually shows you how many times you have to do this particular reaction in the pathway to make it into a balanced pathway. Okay, now, so we're gonna take a look at Calvin cycle in detail. Okay, Calvin cycle, it all starts with this guy, ribulose biphosphate. I blocked it, sorry. So it's ribulose biphosphate. Okay, ribulose biphosphate is a metabolite in which pathway we have discussed? PPP, okay, the pentose phosphate pathway. Previously we saw that ribulose biphosphate is found in PPP for, animal cells or E. coli. So it's part of the pentose phosphate pathway. Okay, so this guy is there. So the first thing that it's going to do is it's going to be activated using ATP, okay, so that we make ribulose 1,5-bisphosphate. A lot of times we'll just call it RUBP. And this guy obviously RU5P. Okay? Don't get confused with, a lot of times you'll see R5P, that means ribose, okay? So we don't want to be confused with that. So we say RU5P for ribulose and RUBP for ribulose 1,5-bisphosphate, okay? Now, RUBP, this guy is a 5-carbon metabolite, okay? And you will use it and you add a CO2 right here, okay? Such that you will take 1 carbon plus 5 carbon. Supposedly you have 6 carbon, right? But what this enzyme does is it's going to split ribulose 1,5-bisphosphate, okay, so RUBP, and CO2 into 2 molecules of 3PG. Where have you seen 3PG before? Glycolysis, okay? It's an intermediate in glycolysis, okay? So 3PG, all right? And then what you have to do then is you're going to activate 3PG, okay, into, what is this? 1,3-BPG, another intermediate in glycolysis, right? And then 1,3-BPG can then become, this guy I usually call it G3P, right? So basically this portion is what? Gluconeogenesis, right? That's all that is, gluconeogenesis. Okay? Now, what I want you to notice is that, let's say here we have 3 RU5P, okay? So we do this reaction 3 times, okay? So then you have 3 times 5, you have a total of 15 carbon, okay? Down here, you have 15 carbon, right? And then you take this reaction 1, 2, 3 times. Now you have a total of, okay, 6 times 3 carbon, so you have a total of 18 carbon right here, okay? Now, in order for this process to occur again, what you need to do is you need to somehow generate the 3 times 5 carbon, the 15, right? Maybe okay? Okay? So that means that if I do this reaction 3 times, I can fix 3 CO2 into 1 3-carbon metabolite right here, okay? And basically, you take this 18 carbons, you activate, okay, you reduce. By the time you get to G3P, you still have 6 times 3 carbon, which gives you 18 carbons total, right? However, remember that 15 carbon has to recycle back for doing this process again, okay? So basically, 15 carbon out of the 18 has to recycle. That means that you only get 1 of the 3 carbons for whatever that you want to do, okay? So it turns out 5 of the G3P will recycle into the entire process and make ribulose biphosphate, okay? 1, 2, 3, 4, 5, okay? 5 of them will recycle, and the 6th one, okay, can be used for whatever that you want to do, okay? And look around here, okay? So we know that you get G3P. G3P is the same thing as DHAP, doesn't matter, right? They can interconvert using which enzyme? Aldolase? No, 他们 interconvert 用什么 enzyme? Trials. 他们是做什么 reaction? Isomerization reaction, right? So 他们是 trials isomerase, right? So you use trials isomerase, you can interconvert G3P and DHAP. So for our purpose, G3P and DHAP, they're pretty much the same thing because the interconversion between those two energy is pretty low, so it's fine, okay? Now, if you have G3P, you can pretty much make anything you want, right? You can go to glucose, it can go down to pyruvate, pyruvate can go down to acetyl-CoA. So pretty much anything you need, you can make using G3P or DHAP. Does that make sense? That as long as your cell can produce G3P or DHAP, it can make everything else. Okay? It can make, it can go down to pyruvate, it can go up to glucose, and then pyruvate can go down to acetyl-CoA to make other stuff. Okay? So as long as you have intermediates in glycolysis, we know from gluconeogenesis that they can all interconvert. Okay, so that's no problem, right? So basically, you converted 3 CO2 into 1 G3P. Okay, so we have C3, okay, and then you have to recycle 5 of the C3 into 3 C5s. Okay, so you want to convert 5 C3 to 3 C5s. Okay, so this, does it sound kind of familiar? Something that we probably touched upon when we were talking about the pentose phosphate pathway during the first third of our class, right? And on your exam, I asked you how to convert C5 to C6 without carbon lost, or C6 to C5 without carbon lost, right? So this is going to be using the carbon rearrangement part of the reaction, okay, of the pentose phosphate pathway. So from here, we need to get it back to here, okay? So we're going to split 15 carbons for recycle, and then 3 carbons for use. And why 15 carbon? Because you have 3 times C5 is equal to 15 carbon. Okay? So this guy and here, they have to match. Okay? So basically, the next phase is about how you convert sugars into sugars. Okay? So guess what? This is all just the pentose phosphate pathway. Okay, now let's start with 2C3. We have to first make a C6. Okay, so C3 plus C3, okay, can give you C6. This guy is aldolase, okay? And then C6 can then, because this guy is fructose 1,6 bisphosphate, okay, F16BP, and we don't need to have a biphosphate. So we can do a phosphatase, okay? This guy is called what? Do you guys remember? In gluconeogenesis, this is something phosphatase, right? Okay? So it's a phosphatase that will remove the phosphate, okay? And you generate F6P, right? Now F6P can then react with another C3, another, in this case, G3P, okay? And you get E4P and X5P, remember that? And one of the X5P can then do its isomerization to ribulose biphosphate, okay? So you got 1 C5, check, okay? Now you're left with a C4, okay? You need to then convert C4 to a C5. To do that, we give it another C3, and we can make C7, okay? And guess what? This reaction is pretty much the same thing, this part, and this part, pretty much the same thing, right? One is 3 plus 3, and the other one is just 3 plus 4, okay? And both of them will generate a sugar biphosphate or bisphosphate, okay? So here you have a S17BP, okay, this guy, okay? That guy is S17BP. So in order to further do rearrangement with it, we turn it, okay, we do another phosphatase reaction, and we turn it into S7P, right? S7P is a 7 carbon, and guess what? 7 plus another 3 can give you 2 C5. This is the second C5, this is the third C5, okay? So we have 1 C5, 2 C5, 3 C5, okay? And those C5s can then all become RU5P, okay, ribulose biphosphate, and then they can do this process again. Okay, so this is the, I don't know, CO2 fixation. And then this part is gluconeogenesis. And this entire part is basically, again, carbon rearrangement. Okay? So this is the Calvin cycle. Okay? So Calvin cycle, it's actually, it looks complicated, but in reality, it's not that complicated. Just as long as you remember the reactants for Calvin cycle is that you have to have C5. Okay, it's a C5 plus CO2 that gives you 2C3. Okay, but in order to regenerate the C5, the C3 has to somehow become C5 again. Okay, so that you have this messy carbon rearrangement phase. Okay, so I really like this particular figure because it really shows you that in one CO2, not one CO2 physician, one G3P production, okay, to produce one G3P, you need to have 3 reactions of this first kinase, okay? You need to have 3 reactions of the CO2 fixation. We'll discuss about this particular enzyme in detail, okay? And then you need to have 6 reactions of this phosphoglycerate kinase, okay? This is gluconeogenesis. So we need to have this reaction 6 times, and subsequently the G3P dehydrogenase, gap dehydrogenase, you need to have this reaction 6 times, okay? So 6 times, 6 times, and then once you get down to here, you'll have 6 molecules of C3, and you only need to spend 5 of those molecules to generate back the 3 C5s. And then that leaves you with 1 C3 for whatever you want to do, okay? For making amino acids, nucleic acids, or whatever, okay? So for the subsequent reactions, they only have to happen once, okay? Because only one arrow, okay? They only have to happen once, and then you can get to your ribulose biphosphate, okay? So this is the overview of Calvin cycle, okay? Carbon rearrangement. So you start with 3 C1s, you end up with 1 C3, okay? So, you know, if I cleaned it, that's the picture of Calvin cycle. So we're going to discuss these in detail. We're going to first discuss about this enzyme and then this enzyme. These we're not going to discuss anymore. They're just gluconeogenesis, you should know. And these, we're not really going to discuss them in detail because they're just pentose phosphate pathway that you should know, okay? So in the carbon rearrangement phase, which enzymes have you not seen before? Or which reactions have you not seen before? Do you guys recognize? Let's go through them one by one. Have you seen this reaction? You have, okay? This is in where? 这个 reaction 是什么 pathway 里面的 ？G3P 加 DHAP 变成 fructose 1,6 bisphosphate 哦。什么东西？这是什么 pathway? Gluconeogenesis. Thank you. Right? How about this one? Same thing, right? Gluconeogenesis. Okay. So this one is gluconeogenesis, and then this is one also gluconeogenesis. Okay. Same thing. And then fructose 6-phosphate. Right. So F6P plus G3P. Okay. Gives you E4P plus xylulose biphosphate. This one is found where? Pentose phosphate pathway. So this guy is in PPP. We've seen this before. Okay? And then E4P plus DHAP gives you cetoheptulose 1,7 bisphosphate. Have you seen this before? No. Okay, this one you haven't seen, right? But like I said, it's pretty much the same reaction as generating F16BP. Okay, pretty much the same thing. Right, so if you haven't seen this guy, you haven't seen this guy. Okay, so these guys are, no, so maybe I should like, so these two you haven't seen. Okay, and then S7P plus G3P gives you R5P and Ribulose Biphosphate. This guy you have seen, maybe, where? Pentose phosphate pathway. This guy also PPP. And then all of the isomerization reactions, also PPP, right? So the only enzyme that you are not familiar with is the aldolase here and a phosphatase here. But essentially, they are pretty much the same reaction as the gluconeogenesis version. Instead of 3 plus 3, you have a 3 plus 4. Okay, so pretty much the same thing. You shouldn't be too anxious about looking at this crazy pathway and be like, whoa, what's he doing? Okay, so all you need to remember is that we're converting 5C3 into here 3 C5s. Okay, so basically this is the bottom line for the carbon rearrangement. Okay? All right. So the first reaction is the preparation of CO2 acceptor, RUBP. So in order to prepare RUBP, you're going to activate RU5P into RUBP, okay? So into the bisphosphate. Well, this is pretty similar because it's fairly similar to what we previously have seen with, for example, where have you seen a something something phosphate becoming a something something bisphosphate? Huh? Glycolysis, right? Which enzyme? Which step? F6P 变 Fe6BP 对不对？第二个 reaction 吗？是吗？第三个 reaction， right？ 第三个 reaction, okay? So if you take a look, remember fructose is a what sugar? Aldose or ketose? Ketose, right? Fructose is a ketose. And ribulose is also a ketose, right? So anytime you see this UL in the middle, typically means it's a ketose, right? We talked about that last semester. Not that you will remember, but we did talk about that, okay? So for example, this guy looks just like fructose, 6-phosphate, except it's 5 carbon, so it's called ribulose biphosphate. So obviously you can pretty much use very similar enzyme to activate it, okay? You can activate this OH into a phosphate, okay? This enzyme would be called what? If you have to name this enzyme, what would it be called? It's a kinase. So it's a kinase, okay? And then, you know, probably like R5P kinase, something like that, okay? If you have to guess the name. I forgot, I should have wrote the name down here, but I didn't, okay? But it's a kinase, okay? And it does just like glycolysis, you turn F6P into F16BP. Similar reaction, okay? So mechanism, same thing, okay? Remember the mechanism for kinases, right? Remember that this guy has lone pairs, right? And the lone pair is going to do something. Do you remember what the lone pair does? No? Probably not, right? Don't really care, right? Yeah, okay, I know. Okay, so that is the first reaction. So we just talked about that. Done, okay? Next reaction is really the key reaction, okay? So this is the, you know, the core of Calvin cycle, okay? Because it's the CO2 fixing reaction. So obviously this is the most important reaction in Calvin cycle, okay? Most important reaction. So you can ignore the stage 1. This is how the book divides the different stages of the Calvin cycle. But stage 1 essentially is the carbon fixation, okay? And the enzyme for this carbon fixation, okay, the enzyme responsible is called Rubisco, okay? It's called Rubisco. The full name for Rubisco is written down here. Okay? It's a long name, okay? Rubisco because it's RUBP, right? This guy is RUBP, okay? Or you can say Rubisphosphate, okay? So Rubis, okay? So Rubis, okay? C and O because it's a carboxylase and an oxygenase. We'll talk about what those mean in just a minute, okay? But this is where Rubisco got its name, okay? Rubisco is a protein that does CO2 fixation, okay? So it's Rubisco. If it can do CO2 fixation, guess what? It's a carboxylase, okay? Remember that we talked about 2 different carboxylases so far. Do you guys remember what they are? Okay, pyruvate 到 oxaloacetate 那个东西叫做那个 enzyme 叫做 pyruvate carboxylase. Okay, so come on, pyruvate carboxylase. Pyruvate becomes oxaloacetate, so it's called pyruvate carboxylase. 还有呢，我们还有讲到一个 enzyme, PEP 到 OAA, right? PEP 也可以到 OAA， 跟 pyruvate 一样。那 PEP carboxylase. Okay, so far we talked about I think only those 2, okay? Those 2 carboxylases, right? So carboxylases tell you that it's an enzyme that can fix CO2 or bicarbonate in those cases, but essentially a CO2, 1 carbon, a fully oxidized carbon, okay? So those are what carboxylases are, okay? So the acceptor molecule for CO2 is this RUBP thing, okay? And we saw in the previous slide that RUBP is the activated product of RU5P, okay? That's fine. And in the process of the dark reaction, what you have is CO2 from the air, okay? CO2 from the air will diffuse into the stroma of chloroplast where it is added at the carbonyl position of RUBP, okay? So carbonyl position, this guy. Oh, we'll see the mechanism in just a bit. Okay, don't worry. Unfortunately, this is a chemistry class, so mechanism, so we will talk about that in case you're like anxious about, yo, where's my mechanism, man? I'm so excited for mechanism. Okay, but obviously no one's going to say that. All right. So for plants, your Calvin cycle occurs in the stroma, okay? And remember where the stroma is, okay? Please differentiate stroma and the lumen. Okay, stroma, lumen. Lumen 是那个 proton gradient 的地方 ，stroma 是它的外面，然后 stroma 的在外面就是 cytoplasm for plants. 可以哈 Okay, so in the stroma you have a bunch of these Rubisco proteins, and the Rubisco proteins are going to do something. Okay, that is fix CO2. Now, Rubisco is probably the most important enzyme in the entire biosphere. We say biosphere is, all the life that we consider this a biosphere, okay? And it's the most important because without Rubisco, you don't have all these sugar and all these bioproducts because we would not have all these organics, okay? So it's really, Rubisco is very, very important, okay? And certainly the most abundant, okay? So this is very interesting. If you were to take the chloroplast and you analyze all proteins in chloroplast, about 15% of all proteins in the chloroplast is Rubisco. Don't forget, in the chloroplast, you still have the photosystems, right? So accounting for all of that, Rubisco accounts for 15%. So you have to understand, one single type of protein, Rubisco, is 15% of the mass, of the total protein mass in chloroplast. It's quite significant, okay? So can you imagine, like, for example, you and 15% is something, okay? It's quite a large amount of something, okay? So don't underestimate how much this is, all right? And the current estimate is that in the entire world, we have about 40 million tons of Rubisco. So 40 million tons is like huge. So then the book gives you a very interesting perspective saying for every living person, you have 20 pounds of Rubisco. 20 pounds, it's about 10 kilo, right? So for every one of you sitting here, you have 10 kilo worth of Rubisco somewhere in this world. Okay, so Rubisco, it's huge. Okay, it's a huge amount of this enzyme just flying around. Okay, and guess what? Rubisco is not limited to only photosynthetic organisms. Okay, so there's soil bacteria, like bacteria in the soil. Okay, they don't do photosynthesis, but they may also have Calvin cycle. For example, in my PhD lab, my lab was working on a system where we can convert electricity into biofuel. Okay, typically you can't do that. Okay, using normal chemistry, you can't do that, but you can do that using bacteria. So one of the bacteria that we use, okay, it can consume hydrogen as the electron source, okay, and CO2 as the carbon source. Right, so it can fix carbon, but it doesn't do photosynthesis. So its carbon fixation pathway is the Calvin cycle. Okay, so it also has Rubisco. Right, so understand that Rubisco, it's a lot. Right? It's like a lot of different organisms have this. Right, and so far we know there are 4 types of Rubisco that have been identified in nature. However, primarily we only talk about one of them. This is the most common type. That is that they have 2 polypeptides, essentially 2 subunits. One is a large subunit and a small subunit. Okay, so typically we call this the RBCL and the RBCS. Okay, so Rubisco the large subunit and the Rubisco small subunit. Okay, so that's Rubisco. All right, the Rubisco reaction. Okay, so we're going to talk a little bit about the mechanism of Rubisco reaction. And again, this guy is RUBP. Okay, so for RUBP, one of the first things that has to happen is that you will have a deprotonation of the alpha hydrogen, or the hydrogen on the alpha carbon. Okay, hydrogen on alpha carbon. Okay, at carbon 3. Okay, I'm not talking about this guy. Okay, this guy is also an alpha carbon. Maybe you can call that alpha prime. I'm not talking about this guy. Okay, I'm obviously talking about this alpha carbon. Okay, so we say that's the carbon 3. Okay, carbon number 3. So you will form this indiolate intermediate. Okay, again, just like at the very beginning of our class, I keep on saying that, you know, you're gonna do something to the carbonyl. Right? It's always doing something to the carbonyl. And always remember that alpha carbon hydrogen, okay, the hydrogen on the alpha carbon can be deprotonated. Okay, other methyl, for example, methyl carbon hydrogen cannot be deprotonated. Okay, so this guy you can deprotonate. And once you deprotonate that, you can form this indiolate intermediate. Okay, and why can we produce this? Why can we deprotonate the proton on the alpha carbon? 有没有人记得为什么我可以把 alpha carbon 身上的 proton 就是拿掉？来嘛，随便丢一个英文字啊 ，R 开头的，还有 resonance. Okay, so remember that this guy, essentially what you can form is right, because this guy you can do that and then it becomes that. Okay, so because it has resonance, so it's resonance stabilized. Okay. All right, so you've got this intermediate. All right, then, okay, now because now you have a bunch of, guess what? You have a bunch of electrons here. Okay, so we removed a proton. That means that the electron is going to be staying around in this region. Okay, so you have a bunch of electrons there. So what you'll do is the electron can then attack something. Okay, so you will form a new bond onto CO2. Okay, because CO2, this guy is a very good electrophile or nucleophile. CO2. CO2 is a good electrophile, right? Right? Because carbon electron is pulled by those two guys, so the carbon is pretty, you know, pretty partial positive kind of. Okay, So then this guy is a good electrophile, so then it's going to attract electrons, so you react, okay? So you put this carbon to this particular carbon there, and in the process, what you'll get is you'll deprotonate the OH right here, okay, the hydrogen right here, so that you form a carbonyl or a keto group right here, okay? And now look, this guy then becomes a carboxy beta keto intermediate, right? So then we're ready to then start to split the molecule. So what we'll do is we'll hydrate, okay? So you have water that can have lone pairs, which then it'll attack a carbonyl center because this is electrophile, this is nucleophile, okay? So you have a nucleophilic attack at the carbonyl center, and then you generate this intermediate, okay? And then what you can have is a collapse back down of this charge. So where do you get the charge? Okay, you get the charge because the carbonyl pop up, okay? Like always, when the carbonyl electron goes onto the oxygen, okay, it's going to come back down. And when it comes back down, you have two choices. You can either move this guy or move this guy. If you move this guy, you generate water. You go back. So obviously this part is reversible. So we don't want to go back. We want to go in this direction, okay, in the top direction. So that's what you do. And you split, you generated the first 3PG, okay? And the other one is right here. However, you still have a minus charge right here. And this guy, all it has to do is grab the proton that the enzyme took from it, and then it also becomes 3PG. Okay? So then at the end you got 2 3PG. Okay? So it all starts with the deprotonation of this guy. Right? So the net reaction is you have a 1 RUBP, which is C5, plus 1 CO2, which is C1, and it can give you 2 3PG, that is C3, and you have 2 of those, which total is 6 carbons, okay, and 6 carbons. Okay, good. So that's the Rubisco reaction. All right, so a little bit about the structure of the Rubisco active site, okay? So obviously Rubisco is a very important enzyme to the entire Earth, I guess, okay? So people are, of course, interested in knowing the Rubisco structure, okay, and different Rubisco structures. So this model is based on the X-ray structure of the spinach Rubisco, okay? Don't ask me why they choose to use the spinach one. Probably just because whoever is doing this did the spinach Rubisco first, okay? Maybe it's easier to purify. And what you see is that you have a magnesium ion here, okay? You have a magnesium ion right here. This guy is the magnesium ion. And this Rubisco model that they got is a complex with the magnesium ion and the transition state analog. So it shows you where the active site is, right? So without a transition state analog, sometimes it's difficult to know where the active site is, okay? So they added something that is similar to RUBP, and in this case it's a 2-carboxy arabinitol, okay? I can't even pronounce that word, right? So this guy is just a transition state analog. You guys remember what transition state analogs are? 记得吗？ Transition state analog 就是一个 chemical 长得跟这个反应的 transition state 很像。那 transition state 如果你长得跟 transition state 很像的话，它会有个什么特性？它跟 enzyme 之间的 affinity 会很强，会非常非常强，对不对？记不记得我们不是说 enzyme 在做的一件事情是它会 stabilize 那个 transition state， 这就是为什么 enzyme 可以降低你的 activation energy 嘛。那 activation energy 一旦被降低以后，你的 reaction 速度才会变快啊。 那它要怎么样子降低 activation energy？ 就是把你的形状，然后把这个 transition state 固定在那边，所以让它就是 stabilize， 对不对？一旦你可以 stabilize transition state， 你就可以降低 activation energy。OK， 那 transition state analog 呢，就是 analog 就是它跟 transition state 长得很像。但是很多时候，其实我们并不一定知道 transition state 长什么样子，所以这个很多是要靠实验以及现在用模拟去 design 去找。OK，but once you know transition state 大概长什么样子，反正你可以去设计一些 transition state 的 analog 去加在这个 enzyme 里面。那它们的 binding 因为很强，然后 binding 很强，但是这个东西又不是它真正的 substrate， 所以它不会被反应掉，所以它就卡在里面。那一旦它卡在里面呢，这个时候你可以再收集大量的这个蛋白 ，OK， 然后去把它变成那个结晶 ，OK，crystallize 它 ，OK。那一旦你得到这个东西的 crystal， 你就可以打 X-rays，OK， 然后你就可以得到它的 structure。Right. So because what they notice is that you have a magnesium ion here, and so that they found that magnesium is involved in the Rubp binding, okay, and activating the water. So as always, magnesium participates in reactions. Okay, it's a metal ion that can sort of assist in reactions. All right. So we talked about this guy, we talked about this guy, and we are very briefly going to talk about these two. Okay, I mean it's the same as gluconeogenesis. Okay, with one exception. Okay, the only exception is that it's using NADPH. Remember in gluconeogenesis, it's the reverse of glycolysis. So glycolysis generates NADH, so gluconeogenesis probably uses NADH. Okay, but in this case, we're using NADPH. And what's the reason for that? I mean, come on, this is a photosynthetic organism, right? The net reaction from light reaction, the net product from light reaction is NADPH. So, I mean, come on, the evolution, you know, you got like, oh, I generate NADPH. Of course, you're going to evolve an enzyme that uses NADPH over NADH. Okay. 有没有人听不太懂我在讲什么？肯定有，对不对？有，对不对？有吗？吼，好来，没有了。这边要跟你讲说，我刚不是说这一段吗？这一段是 gluconeogenesis， 对不对？但是 gluconeogenesis 啊，它 ATP 是 ATP， 但是 gluconeogenesis 这边不是 NADPH 啊，大部分是 NADH 啊，对不对？因为那个你的那个 gap D 啊，就是这个 dehydrogenase 在 cytosol 里面大部分是 generate NADH， 对不对？我们在说 glycolysis 的时候，但是在 Calvin Cycle 里面呢，它用的是 NADPH，OK， 它用的是 NADPH。那我的问题是，它为什么要用 NADPH 而不是 NADH 呢？为什么要不一样 ？I mean， 原因就很简单啊，原因是因为我这个植物或者是我的蓝绿藻，它光合作用 light reaction 结束的时候出来的是 NADPH 嘛，所以在这些细菌或者这些生物里面，我的 NADPH 的量会比较多。OK， 那在 evolution 的过程中，我当然我 evolve 出的合成性的代谢途径也都会是使用 NADPH 的。Make sense? Kind of. Okay. So the only exception is it's going to use NADPH, right? Instead of NADH. That's the only exception. And then this guy, same thing. Okay. All right. So the Calvin cycle key concepts here is that you have Rubisco, this guy, okay? So you got Ru15BP plus 3CO2. You run it 3 times, like I said. And once you get to 3PG, you take 3PG to G3P. You run it 6 times, okay? So, okay, total, what you get is 3 CO2, okay, plus 9 ATP, 3 right here, and 6 right here, plus 6 NADPH right here, and you generate 1 G3P. Okay, so the core reaction for Calvin cycle happens pretty much right here. So the part that has to deal with energy equivalence happens all right here. Okay? So after this part, everything else is just rearrangement. Okay? So the net reaction occurs from the part on the right here. Okay? So net reaction occurs right here. 3 ATP, 6 ATP, so total 9 ATP, 3 CO2s, and 6 NADPH. Done. And then this is your product, DHAP or G3P. Okay? So this is the net reaction for photosynthesis all. Right? Right? And G3P is the same thing as DHAP. Okay? Alright, now we're going to talk about the carbon rearrangement phase. Right? So a lot of times people will call it stage 2, okay, the regeneration of the acceptor. Right? And really, to complete the Calvin cycle, it is necessary to generate enough RUBP to keep the cycle going. Okay? And RUBP is derived from Ru5P. Okay, so we need to generate Ru5P. This means that we need to regenerate 3 moles of Ru5P for every 3 moles of CO2 fixed, like what I said. And this is accomplished by the regenerative phase. And basically, I like to call it just the carbon rearrangement. Okay? So to generate 3 moles of RUBP, 15 carbons, we need to have 5 moles of G3P, okay, which is 15 carbons. Okay? And of the 5 moles of C3, 2 of them are DHAP, 3 of them are G3P. Okay? So then the net reaction of this entire phase, okay, of this entire phase of the Calvin cycle, the net reaction is this. 2 DHAP plus 3 G3P gives you 3 Ru5P. It does not require ATP, it does not produce ATP, and it has nothing to do with NADH, NADPH, that kind of stuff. Absolutely nothing. It's just carbon rearrangement. Okay? All it ever does is rearranging carbons. 3 plus 3, 3 plus 4, 7 plus 3 gives you 2 5s, etc. Like what we talked about at the beginning of our class. Okay? Alright, so 2C3, 3C3, 3C5. Okay? Pretty straightforward. Alright? So I'm going to talk about 2 figures from your book. Okay? You know, I want to explain it a little bit before you get too confused and was like, well, what do you mean? Like, you show me this picture and this picture looks okay, but then the book shows you this, and you're like, well, what? Okay? Okay? So this is the regenerative phase, and this figure is actually, if you look at it like this, you're like, what? Okay? That's at least the first feeling that I got. I was like, wait, what? This is not a circular thing, right? Okay, look. This thing is this is talking about the regenerative phase, okay? And this is it's actually a good figure, okay? Don't get me wrong. So, understanding that our reactants are 5C3. 1, 2, 3, 4, 5, okay? Products are 3 C5s. 1, 2, 3, okay? So, you have C3 number 1, number 2, number 3, 4, and 5. And then this is C5, 1, 2, and then 3. Okay? And, you know, in a way, this is actually telling you going from the top to bottom, the sequence of reaction. Okay? Just like in the circular figure, this is a sequence reaction. Okay? Sequence of reaction. So the first is G3P plus DHAP, and essentially goes down to fructose 6-phosphate. Obviously, it's missing the fructose 1,6 bisphosphate. Okay, so you have DHAP plus G3P. It gives you F6P. Okay, F6P, and then you added another G3P. You get 1 C5. Okay, this is the first C5, and then E4P right here. You give it another 3 carbons. It becomes a 7 carbon. Okay, 7 carbon. and then 7 carbon adds another 3, gives you 2, 5. Okay, so pretty much the same thing that we talked about using the circular figure, but this way it sort of just helps you to really visualize where are the 3 carbons coming in and where are the 5 carbons leaving. Okay, so you have a total of 5 C3, and then you leave with 3 C5. Okay, and please pay attention to the enzyme's name. Okay, Aldolase you should know, and this guy is a transketolase from the PPP you should know, right? This one you haven't seen before, so it's called the transaldolase. Okay, transaldolase. Okay, transaldolase. Have you seen that before? You did. What I'm saying, you did. PPP, right? You saw the same thing in PPP. Yeah. Transketolase and transaldolase. And then when you get down to here, phosphatases, same thing here. You also have a phosphatase, and then you have another transketolase. So all of these enzymes you should be familiar with, okay? Yeah, so transaldolase, transketolase. So those are the 2 enzymes really used in this process, okay? Right? I'm going to conveniently skip over this because if you were to read this figure or look at this figure from your book, you're going to again be like, what? Okay, all right? But can I just very simply summarize what this figure is trying to tell you? Okay? What this figure is trying to tell you is that, look, 3 CO2 plus 9 ATP plus how many NADH? NADPH? 6 NADPH gives you what? G3P, right? Right? And remember, you need to have 2 G3P go to glucose, okay? 2 G3P goes to 1 glucose without needing anything, 对不对？就是2个 G3P 回到1个 glucose， 你不需要再多吃 ATP 或者是 NADPH 或者是 anything，right？ G3P 上面是什么？就是 F16BP 了嘛 ，OK？F16BP 然后变成 F6P 不消耗能量 ，F6P 变 G6P 不消耗能量 ，G6P 变成 glucose 不消耗能量 ，OK？ 所以这边是 energy neutral，OK？ 这边不会吃任何 energy。So what this figure is trying to tell you is to make 1 glucose, you need to have 6 CO2 plus 18 ATP plus 12 NADPH. Okay? Right. Basically, 6 ATP here plus 12 right here. That gives you 18. And then 12 NADPH right here. And then 2 G3P. G3P goes to glucose, no problem. Okay? And of course, if you want to take glucose, make amylose. Do you guys remember what amylose is? 有没有记得 amylose 是什么？淀粉类的那个东西嘛，对不对？OK。然后，但是如果你要把 glucose 变成这个 amylose 的话，你还是需要再吃一个 ATP， 就是 look at this part. G6P, G1P, ATP, okay, and then it becomes amylose. Okay? So this is how you go from CO2 to carbohydrates. Okay, 6 carbon carbohydrates. Alright, 所以我们讲这个讲完之后就 pretty much this is photosynthesis. Okay, this is photosynthesis. Alright, recall light reaction. Okay, we can take 8 photons and make 2 NADPH and 3 ATP. Okay, please remember this. Okay, please remember this. And it just so happens that we saw in the dark reaction, for every 1 G3P you want to produce, you need to have 3 CO2. Of course, this one is C3, this is C1. Okay, you need to have 3 of these. Okay, and then turns out you need to have 6 NADPH and 9 ATP. And guess what? It just so happens that it's these 2 times 3. okay? It just so happens that they balance. It's like magic, okay? It just so happens they balance, right? So the light reaction and the dark reaction turns out to balance, okay? The net product of the light reaction, this guy and this guy, is used to make this guy and this guy, okay? Right? So if you go from the light reaction to the dark reaction, you will not net produce something. For example, you don't net produce ATP, you don't net produce NADPH, okay? It's pretty balanced, okay, in this entire process. But of course, remember that if you have too much light and not enough CO2, you cannot consume these two guys, so you will continually consuming ATP while NADPH is not consumed, okay? Remember we talked about that when we talked about cyclic electron flow, right? When you don't have enough CO2, you will not consume the electrons, but your cellular function still requires ATP, okay? So you can use cyclic electron flow. So at the very, very end, we can say 24 photons equals 1 G3P. Okay, so this is the sort of the little conclusion about how much of something you can produce per how much amount of photon. How many photons do you need to make glucose? 48, okay, 48 photons, right, 48 photons, right? Okay, so photosynthesis, I think we should be good with this. So again, always definitely the most important part for my purpose, okay, is you should understand how many photons to what, you know, to how many NADH, how many ATP, and then how many NADPH and ATP is required to synthesize G3P, that kind of stuff. Okay, so the process and the pathway, you should know all of that. Okay.